बिस्मिल्लाहिर्रहमानिर्रहीम अस्सलामु अलैकुम व रहमतुल्लाह। আজকে আমরা এপিডেমিওলজি অফ ইনফেকশাস ডিজিজ এটা নিয়ে আলোচনা করতে চাই। বর্তমান কোভিড সিচুয়েশনে এটা সবার জন্য ইম্পর্টেন্ট, বিশেষ করে যারা পাবলিক হেলথ, মাইক্রোবায়োলজি কিংবা যারা নিপসামের স্টুডেন্ট, তাদের জন্য এই লেকচারটা তৈরি করা হয়েছিল। এপিডেমিওলজি অফ ইনফেকশাস ডিজিজেস লেকচারটি তৈরি করা হয়েছিল 2018 সালে, যখন আমি নিপসামে মাইক্রোবায়োলজির প্রফেসর অফ মাইক্রোবায়োলজি ছিলাম। আমাদের এই লেকচারের লেসন প্ল্যান: হোয়াট আর দ্য কম্পোনেন্টস অফ ইনফেকশাস ডিজিজ, ইনফেকশাস এজেন্ট, human reservoirs, carrier and importance of carriers, animal reservoir, inanimate reservoir, source of infection, route of infection, host factors, control of epidemic disease, that is establishing a program, and surveillance. Infection is different from other medical and pathology disciplines because infection, it spreads. Infectious disease spreads. Other disease doesn't spread. What are the components of communicable disease? Characterized by three components. It must have a living infectious agent. There must be suitable host and suitable environment. So, three components. Infectious agent, you all know, infectious agent play a very important role, and it includes viruses, bacteria, protozoa, fungi, and helminths. To establish infection, the infectious agent must have the capability to multiply, emerge from the host, reach a new host, and infect the new host. So, knowledge of the mechanism that the organism uses at each of these four stages may help in identifying the most vulnerable stage at which to direct the control measures. The ability of the infective agent to survive in the environment is an important factor in the epidemiology of infection. Reservoir and source of infection. Reservoir, specific ecological niche upon which it depends for its survival, and it may be human, animal, or non-living. In case of gonorrhea, syphilis, HIV, measles, human is the reservoir. For zoonotic disease, vertebrate animals are reservoirs, and in some diseases, soil and water, non-living sources are reservoirs like tetanus, botulism, etc. Source is a medium through which infectious agent transmits from a reservoir to the host or to another reservoir, but it cannot survive indefinitely. So the difference between reservoir and the source should be clear to everybody. Human, for example, human is the reservoir of Salmonella typhi, but contaminated water, milk, and other food products are source for Salmonella typhi. Human reservoirs also, sometimes there are carriers. Carriers means who harbors the infective agent without showing the signs of the disease, but is capable of transmitting the agent to other person. Carrier may be healthy carrier, convalescent carrier, chronic carrier, or incubatory carrier. And in the COVID situation, you can understand there are many healthy carrier, asymptomatic carrier, incubatory carrier. Convalescent carrier can also be present, and chronic carrier, the classical example is hepatitis B and hepatitis C. Importance of carriers. Carriers play an important role in the epidemiology of certain infections like poliomyelitis, meningococcal meningitis, typhoid, and amoebiasis. Carriers also play an important role in COVID. What are the regions? because large number of carriers than the patients. Number of carriers are always more than the patients. Healthy carriers and their contacts are not aware of the infection, so they can easily transmit it to others. And chronic carriers may serve as a source of infection for a long time. How the disease agents are transmitted from animal region? There are 4 ways. Humans use the animal for food, for example, teniasis. Vector, transmitting the infection from animal to human, for example, plague, mosquito, plague by flea, and viral encephalitis by mosquito. Animal bites the human being, for example, rabies. And animal contaminates human environment, including food, salmonellosis. These are the methods of transmission from animal region. What are the routes of transmission? It is known to most of us. There are 5 routes of transmission, of which direct, penetration of skin, inhalation, ingestion, transplacental. Contact, it can be through direct contact, through fomite, close intimate contact, or sexual contact. Penetration of skin can occur directly by the organism, like hookworm, through the bite of vector, malaria. Inhalation of the air, airborne infection, and droplet infection also by inhalation of air. Ingestion from contaminated hands, food, and water. And some disease can be transmitted by transplacental route, HIV, syphilis, toxoplasmosis. Some agents may be transmitted by more than one route, plague, by flea, and inhalation. Host factors. For a disease to establish, there are 2 factors, agent and host factors. Host factors includes the occurrence and outcome of the infection are in part determined by the host factors. And the term immunity is used to describe the ability of the host to resist infection. And immune response also modify the nature of the pathological reaction to infection. And allergic reactions in response to infection may significantly contribute to the clinical and pathological reactions. So you see, host factors are very important for the outcome of infection. You can take the example of COVID infection. Usually, you see, not that much manifested in the neonates, children, and it is have less morbidity in younger generation, but more morbidity in older people with comorbidity. So host factors, that is immunity, may be non-specific. You all know physical, chemical, biological factors, skin, bone, mucous membrane, cough, other reflexes. Can be specific, genetic factors like, and acquired factors may be active, natural, passive, natural, active, artificial, passive, artificial. You all know these terms. Active, natural by clinical or subclinical infection. Passive, natural from mother to fetus. Active, artificial by vaccination. Passive, artificial by preformed immunoglobulins. Factors affecting the host immunity are age, sex, pregnancy, trauma, fatigue, and herd immunity. You can see in the COVID situation is how it is affecting the host immunity. Sex also, male predominance, pregnancy. Trauma, fatigue also have role, and herd immunity. Herd immunity, you know, if 70% of the population is either clinically infected or by vaccinated artificially, rest of the population that's free from the risk of infection. This is herd immunity. Let us discuss about the control of the communicable disease. For control of a communicable disease, we need to establish a program. What is a program? A program is based on detailed knowledge of the epidemiology of the infection and on effective public health organization to plan execute, and evaluate the project. Plan, execute, and evaluate. The program must include some mechanism for recognizing the infection and confirmation of the diagnosis. Recognizing the infection, that means they will define the criteria for case definition, we say, and then to confirm the diagnosis by some laboratory test. And second component is must notify the disease to appropriate authority like IDCR or government, finding the source of infection. And the source of infection can be found by many different ways, like contact tracing, by finding cases, carriers, etc. And assessing the extent of outbreak by finding other cases and exposed persons. So the components, if we discuss in detail: recognition by clinical sign symptom, lab confirmation, notifying to the appropriate authority depending on the severity of the disease, source based on by contact tracing, and extent about by finding other cases and exposed person. And these are all important for successful program to control the communicable disease or infectious disease. Incubation period is very important to control the disease because it's of great value in finding the source of infection. For example, in case of gonorrhea, source of a case of patient should list all sexual conduct in the preceding 5 days before the onset of symptom. But in the case of syphilis, the suspect should include all sexual partners in the 3-month period preceding the appearance of chancre. So here, the incubation period is helping how to detect the cases and contacts. Knowledge of incubation period is also helpful in identifying whether an outbreak has resulted from point source or from multiple sources. And it is also required for protection by passive versus active immunization. If it is of long period, then active immunization can be started. But if incubation period is very short, then passive immunization must be started. Assessment of the extent of the outbreak. It involves finding other infected persons in addition to notified, identifying others who have been exposed to infection, like contacts of known patients who have been exposed to a common source. And what are the methods of control? There are 3 main methods for control of the communicable disease. The methods are elimination of reservoir of infection. This is very important. If there is any reservoir, interrupt the pathway of transmission, break the pathway, and protect the susceptible host. And sometimes all 3 methods are applied together for effective control. Elimination of reservoir, say if it is a human reservoir, find and treat all infected person, isolation of the patient, and quarantine of the patient. All these 3 things we are practicing for COVID patients. We are finding and treating all infected person, isolation of the patient, and quarantine. So this is a classical example. Animal reservoir, if there is any animal reservoir, then appropriate action will depend on usefulness of the animal, how intimately they are associated with the human being, and feasibility of protecting susceptible animal. This is, you can see the rabies cases. If the dog is very useful, then you will have to vaccinate the dogs. If not that useful, previously you can kill the dogs or eliminate like this. So regarding animal reservoir, you will have the action will be determined by the things, how important the animals are, where they live. In case of non-living reservoir, like just you can take the protective measures like tetanus, is just you can by active immunization or by avoiding the source. Interruption of transmission mostly involves improvement of environmental sanitation. Example is cholera or diarrheal diseases. Improvement of personal hygiene. You see in COVID situation, hand hygiene is so important. Control of vectors by alterations in the environment and use of pesticidal agents used in malaria, and protection of the susceptible host can be achieved by active immunization, passive immunization, use of antimicrobial drugs, and mass campaigns. Active immunization when the vaccine is available. Passive immunization, you also know, when the incubation period is very short. Use of antimicrobial drugs, chemoprophylaxis for prevention of malaria, meningococcal meningitis, and bacillus dysentery. Mass campaigns indicated for dealing with acute epidemics as a method of controlling or eradicating epidemic disease. Like you see during this COVID situation, mass campaigning is widely used. When any vaccine or drug used for mass campaign, it must be effective, safe, cheap, and simple to apply. And following the emergency operation of a mass campaign, the program should be integrated into basic health services of the community. Any artificial immunization in control of infection, passive immunization, immunoglobulin against chickenpox, HIV, active immunization, childhood immunization, and human papillomavirus vaccination for women, active passive immunization. Smallpox has been eradicated, polio also, diphtheria, tetanus, and measles are controlled. So the use of drugs in the control of infections, aim of mass treatment. When we use drug for mass treatment, they protect the uninfected individual, they arrest the progression of the disease and they reverse the pathological damage, they eliminate infection and thereby prevent further transmission of the disease. So mass treatment is sometimes very helpful, and classical example in our country is use of albendazole for control of filarial infection. Drugs can be used in three forms: chemotherapy, chemoprophylaxis, and chemosuppression. Chemotherapy for treatment of sick individuals, chemoprophylaxis, protection of persons who are at risk, that like during traveling to endemic area. Chemosuppression, prevention of severe clinical manifestation and complication in infected persons. Strategy of drug use: mass chemotherapy used in albendazole for filaria, penicillin for use. Selective population therapy, treatment of all persons that are found to be infected at initial and subsequent service. For example, in our country, anti-helminthics are used for school-going children. Targeted chemotherapy, treating only those individuals harboring heavy infections or high-risk groups. Treatment of persons aged 5 to 20 years of schistosoma hematobium infection. So there are three types of chemotherapy: mass chemotherapy for all, selective only for who are found to be infected initial and subsequent service, and targeted chemotherapy. Various strategies for large-scale use of drugs are being applied to trachoma, schistosomiasis, malaria, and onchocerciasis. Drug specification for large scale: the drug must be effective, highly effective, without development of resistance, must be safe, no dangerous side effects, and wide margin between therapeutic and toxic dose. Regimen should be simple, like single dose oral, acceptable without any unpresentable side effect, affordable, or should be free. So antimicrobial resistance become a serious problem, serious public health concern. There is resistance to first-line drug of TB ranges from 2 to 40% in many countries. MDR and XDR TB are increasing. 90% cases of Shigella dysentery are resistant to cotrim, nalidixic acid. Multi-resistant Salmonella typhi is widespread in endemic countries. Chloroquine-resistant malaria is global. There is ESBL, MRSA, and VRE. So factors for development of resistance: overuse and misuse by doctors and health personnel, poor compliance, self-medication, counterfeit drugs, poor control of antimicrobials in the hospitals. and poor standards of hygiene like personal and environmental in hospitals, and use of antibiotics in animal husbandry, horticulture, and aquaculture. All these are responsible factors that contribute development of resistance. Unethical promotion by pharmaceuticals is also important for development of that resistance. Surveillance of disease. Surveillance of disease means exercise of continuous scrutiny and watchfulness over the distribution and spread of infections and the related factors with sufficient accuracy and completeness to provide the basis for effective control. And it has got components like systematic collection of all relevant data, orderly consolidation and evaluation of this data, and prompt dissemination of the results. Objective of the surveillance, first objective is the recognition of acute problem that demand immediate action. For example, this outbreak, COVID outbreak. Second objective is to provide a broad assessment of the specific problem in order to discern long-term trend and epidemiological pattern, to guide and monitor intervention, and finally to assess their impact. Thus, surveillance provides a specific basis for ascertaining the major public health problems in the area, thereby serving as a guide for planning, implementation, and assessment of the program to control the communicable disease. And this is how the epidemiological data are gathered. First, you can registration of death. You can see your just COVID example, registration of death. We are registering the death. Notification of the disease and laboratory epidemics, laboratory investigation, data from routine screening, investigation of individual case and epidemiological survey, data from different clinic, health center, hospital, other service institution, and non-medical statistics sometimes also help useful demographic and environmental data. All these data are sources of epidemiological data in surveillance of disease. So thank you very much for your patience hearing.